Nick Jackomis, and today my guest is Dr. Dirk Hoffmeister. Professor Hoffmeister is a pharmaceutical microbiologist at the Hans Knöll Institute in Germany. His background is in both botany and mycology, the study of fungi, and his lab studies various aspects of fungal biochemistry and molecular biology, including the biochemistry and molecular genetics of psilocybin production in magic mushrooms. We spoke about various topics in mycology, mostly related to Psilocybe mushrooms. This included how psilocybin is synthesized from the amino acid tryptophan, the ecological reasons for why some mushrooms produce psilocybin, why magic mushrooms turn a vibrant blue color when they are physically damaged, the production of monoamine oxidase inhibitors, or MAOIs, by certain species of magic mushrooms, and the ecology of psilocybin mushrooms. We also touched on some of the other areas of mycology that his lab studies, so if you're interested in mushrooms in general, and magic mushrooms in particular, and psilocybin, and how it's produced, and sort of all of the inner molecular workings surrounding how that happens, this will be a great episode for you. Dr. Hoffmeister is one of the world's experts on the biochemistry of this stuff, so I learned a lot of really cool things from him. As always, if you enjoy the content I'm producing on the Mind and Matter podcast, please like, share, and subscribe. You can sign up for my free weekly newsletter at mindandmatter.substack.com. On that newsletter, I'll send out every week. You will see who the upcoming guests and topics of discussion will be. You'll see some of the research that I'm reading related to the topics I cover on the show and what I think about that. And I also post interesting links to other things that I'm reading or other content I'm producing elsewhere on other channels. You can also support the podcast by looking at the links in the episode description or in that newsletter. You'll find some links with discount codes to some products that I like and that I use. And by using those products through that link, you'll directly support the podcast to help me keep it going and keep it growing. This episode is supported in part by the Amino Company. They specialize in making science-backed amino acid products that you can mix into any drink. Their products contain a mixture of essential amino acids, the building blocks of proteins in the body, as well as other nutrients including minerals like iron and electrolytes like potassium. Your body is constantly repairing damage, and your muscles and tissues need the right mix of amino acids and nutrients to do this effectively. One thing I like about Amino Co. is they actually conduct clinical trials to determine what their products really do. They have a variety of formulations engineered for different purposes, and my personal favorite is one called Heal, which has been shown to be 3 times more efficient at triggering muscle growth and repair than other protein sources. It helps maintain healthy inflammation levels and preserve muscle mass during periods of inactivity. I mix this product into the water bottle I bring to the gym and consume it before, during, and after my workouts, and I have felt a noticeable difference in my performance during those workouts and my recovery times from soreness and fatigue afterwards. Their products are keto-friendly, soy-free, vegetarian or vegan, gluten-free, and non-GMO, so they are compatible with almost any diet or lifestyle. You can support the podcast and try Heal or any of their other products by using the discount code MIND when you visit aminoco.com/mind. You will get 30% off your purchase. If you work out regularly or do intensive exercise, I recommend trying AminoCo's products. I get a lot of companies reaching out to me about advertising, and I only end up using and liking a small percentage of the products that I see. So check out aminoco.com/mind and use the code MIND to try these products today for 30% off. Today's show is brought to you in part by Dosist, an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. And with that, here's my conversation with Dr. Dirk Hoffmeister.
Jena, which is pretty much in the center of Germany. Not that it's not that much of a it's too small for a big city, and it's too big for a small town. 100,000 inhabitants, college town, quarter of them being students, and fun city.
Yeah, and you're from Germany originally?
Yes, I am.
Excellent. And can you just kind of give everyone a little bit of a background in terms of what your scientific training is and what you study very, very generally?
Oh, absolutely, sure. So I'm currently a professor of pharmaceutical microbiology, and by training I'm botanist and mycologist. These were my majors, and so basically back then, so 25 years ago, or longer, even 30 years ago, I didn't really, think about the things and the subjects I'm thinking about today. So botany and mycology, this was my training. And I then went on for a PhD degree in basically in bacteriology on an antibiotics related topic, which took me a little bit away from my fungi and from my mushrooms, which I was interested in before. And then later on, after I finished my PhD degree, but I should say, all the time now, from being an undergrad throughout my PhD time as a PhD student, I was interested in small molecules, in all these wonderful molecules that nature came up with as antibiotics, as something that tastes spicy or hot or sweet, or all the various herbs. I mean, just look around in your kitchen. Just think about your kitchen. What's there? What's tasteful? And so nature basically has evolved so many and so wonderful molecules. And this interest was kind of, has been constant all over the years. And so when I was done with my PhD degree, I went to the United States for my postdoc training to the Midwest, Madison, Wisconsin. And there I kind of got reintroduced to fungi. So it basically was kind of a circle I made, and which then took me back to the fungi and their small molecules, their natural products. And so this was about the time when I had to think about my own independent research program. Mm-hmm. I had to, but basically I was kind of decided to follow an academic career, and so I had to think about what kind of niche, what kind of research I would like to do as my own independent program, and noticed that mushrooms are little understood in terms of how they make these natural products. This is the correct term, the technical term, natural products, small molecules, often bioactive. And when you think about mushrooms and what are the most prominent and most well-known molecules, you think about toxins. Mm. And so I thought about I should do research on the death cap, this incredibly toxic mushroom that appears in the fall, and that could even kill you. And so this was one thought I had. The other thought, this was back in 2003, 4-ish. And this was one choice, and the other obvious thing, and I believe this was the reason why you introduced me to your episode today. The other obvious choice were the magic mushrooms, and from today's perspective, I was fortunate to choose neither one nor the other, because the techniques and the methods were not as advanced as they are today, and I would have miserably failed. But anyway, the idea was in my mind, and so I went on with other molecules. Went back to Germany, accepted the junior group leader position, then moved back to the United States, again to the Midwest, even further north to Minnesota, and accepted a faculty position there, a tenure track position there, and then relocated as well to my current place, to expand on that topic and say with more resources available and with the methods being more advanced than they used to be 10, 15 years ago.
Yeah. So what initially drew you to mycology? Mycology's, it's not, it's become sort of famous recently with the whole psilocybin mushroom thing, but it's never, you don't typically, it's not considered a big or sexy field. So how did you get into mushrooms and mycology to begin with? Was this a lifelong interest?
Well, this is kind of an individual perspective, whether it's a sexy topic or not, I guess. Yes, definitely. This is a tough one. What drew me to mycology? This is I think this is a combination of two aspects. First of this, it has to do with a lifelong interest. So as a kid, I grew up in a rural area, a rural place in the far southwest of Germany, and nature was readily available. You know, I just left my parents' home and I was in the forest. Hit the forest and found the mushroom even as a kid, and I knew the most important ones as a kid, and the chanterelles and the bolets and so. So this was I think this was one aspect that caught my interest very early on, and the second the second thing was one of my academic teachers, the most influential one probably of my academic teacher, who now passed away a few years ago, who was such a brilliant, such a talented teacher and researcher. And this kind of, so I just got hooked again after, 15 years as an undergrad. And so I took all the classes, all the courses, and this kind of, laid the foundation of my interest. Although his research back then was completely differently oriented. It was more about evolution and systematics and taxonomy. And whereas my research is more into molecular biology, chemistry, biochemistry. So the direction is a different one, but the basis is the same. It's about mushrooms.
Mm-hmm. Yeah, and from a taxonomic perspective, so you said you majored and you studied early on botany and mycology. Mm-hmm. Where do fungi sit relative to plants and animals in the tree of life?
Much closer to animals. Much. I mean, traditionally the animal biologists had more, to think about more animals than the botanists about plants. So they took mycology to plants. So the tree of life. So if you have the plants are branching here. So all evolutionary biologists who are listening now, please forgive me if I simplify the tree of life with just my 10 fingers. So plants are here basically, and that branch, that direction, then later off branches off here into very simple, in very simple terms now, into fungi and animals.
I see. Yeah. So superficially, a lot of people think of fungi as closer to plants because they kind of grow out of the dirt like plants, and they vaguely look like plants, but they're actually more closely related to us.
They cannot walk away, like animals, or most of them. But we should keep in mind that the relationship between plants and fungi is a very, very close one. It's a coevolution, or has been a coevolution over 500 plus millions of years, probably, even before the basically the plants went out of the sea and conquered the land. And so what we see today is or what it resulted in is that plants and fungi have very close relationships, be it a symbiotic one, so that each partner or multiple partners benefit from each other, or fungi use plants as a host for parasitic fungi use plants as their host, and which is kind of a one-way street now, the benefit. And so the notion of plants and fungi being very close to each other is kind of justified. Not from an evolutionary perspective, but from just going outside and look what's going on in life.
Yeah, so when you look at the genetics and the literal familial relationship, fungi are closer to animals and plants. Nonetheless, they are closely intermingled with plants in terms of their actual like ecological interactions.
Absolutely, absolutely, and they couldn't survive without the other one. I mean, we were talking about fungi now, or mushrooms. And many mushrooms are live in a close symbiosis with look at all the on a global scale, that the northern hemisphere is dominated by forests. I mean, so don't think about deforestation, don't think about humans that destroy habitats, but just leave the planet as it would have developed. Mm-hmm. Without human influence, without human impact. And then the northern hemisphere, in temperate areas, in, let's say that's North America, United States, also in boreal forests, Canada, and further north, or Scandinavia further north in Europe. These are dominated by trees that depend on mushrooms as a partner in the soil, in the ground. They have a very close physical interaction between plant roots and the fungal hyphae, the filamentous cells and of the mushrooms for exchange of nutrition, and so the one couldn't live out without the other one, without the other partner. And this is why we so as we walk through a forest in fall, and see all the various mushrooms that pop up and grow out of the soil, we obviously think about this. They must have something to do with each other. I mean, not from an ecological standpoint, but from an evolutionary standpoint, mm-hmm. as we know now, or many years ago now.
Yeah, and I think when people think about mushrooms, they're thinking of the fruiting body, the thing that actually comes out of the ground from time mm-hmm. to time.
Yes.
But what you're alluding to is there's this vast network underneath the ground.
In the ground, exactly. Yeah.
Can you talk a little bit about what that is? What is the life cycle of a mushroom, and when it's underground, what is that called, and what is that sort of form of the mushroom, and then why do these things periodically make these very, very big fruiting bodies that pop up from time to time?
So the fruiting bodies like this one here, this is just for reproduction. So this is what we see. That's right, Nick. So this is what we see as a how we perceive mushrooms. But this is just a temporary thing, an ephemeral thing. These are formed for reproduction, for sexual reproduction. So the spores, that are ready to grow out and make the next generation, they would be formed here at the cap, and so this happens once in fall. So that there are the fruiting bodies, they appear quick and are there for a couple days. And in the soil, in the ground, there's kind of a web-like structure of long filamentous hyphae, really like filaments, very long, and they are very thin. So much thinner than a tree root. Mm. So in other words, they can access little niches and pores in the soil that a plant couldn't grow into, and so they can take up nutrients and supply the tree with nutrients, and this kind of relation and these hyphae. And the entirety of hyphae would be called mycelium from mucus mushroom or fungus. And so this mycelium stays there, year round, and supplies the tree with elements, with nutrients, phosphorus, nitrogen. Protects so basically it really, if this is a plant root here, and then the mushroom hyphae, these underground cells, they would wrap themselves around the root, and also offer some protection and drought resistance. So this is the benefit that the tree has. The tree in return can make sugars, nutrients, by just taking in CO2, fixate CO2 and reduce it into sugars. This is what we live from, so I will have dinner soon. You will have breakfast maybe. Soon you will eat cornflakes or oat flakes.
My mushroom coffee is all I've had so far.
Okay. And so the benefit the mushroom has is that it has access to nutrients, to sugars, to carbon, carbon source.
Is it fair to say that basically, so these mycelial networks are providing the plants with micronutrients, like certain elements, also physical protection, and I think we'll talk about antibiotics and things like this. And the plant is giving the fungus primarily macronutrients like sugars.
Yes, yeah, that's correct.
Okay, so
and this is the case for a symbiotic relationship. So both partners, in some cases there are multiple partners involved, but keep it simple. Both partners are they benefit from each other. And in case of a parasitic mushroom, there are many out there. They attack a tree. Maybe a limb broke off, or a thunderstorm hit the tree, and so there's kind of an injury, and then the mushroom can enter the tissue, the tree tissue, and can now degrade living matter, living tissue in the tree. And this is a process, whoever is stronger, and that can go on over decades, and eventually the mushroom will win, I would say. And the tree will die, and then serve, and this dead wood will then serve as substrate, also as a macronutrient for new life. Think about how. Think about a forest, maybe somewhere in East Coast or Canada or Minnesota, a forest without any human influence. It just develops over centuries, over millennia, and during that time, many generations of trees will die. They will fall off, or maybe a storm hits a forest, and then they just tip over. But still, after decades and centuries and millennia, the dead wood, the dead trees, they will not reach the sky, but they will have been degraded in the meantime. Why? Because degrading fungi, not the symbiotic ones, but the degrading ones, did their job, and it takes about maybe 100 years for a whole set of mushrooms to degrade a tree, a big, substantial, maybe oak tree that fell off or fell over, to degrade it into powder.
Mm-hmm. So there's a couple different broad types of mushrooms. There's the ones that want the plants to stay alive, to engage in the symbiotic exchange, and the ones that want to degrade them, and they're always in some kind of cyclical balance in the whole ecosystem.
Mm-hmm. It's kind of what scientists call carbon cycling. So we have CO2 in the air as a substrate, as something beneficial for plants, because they live from CO2, carbon dioxide. And so this is what basically trees fixate the CO2, make biomass, they grow. At the best example, at the West Coast, these wonderful sequoia trees. I mean, they just hit the sky, and then degraders need to come in to basically to disintegrate this biomass, this wonderful biomass that we call trees, to disintegrate it again and make it available to new life, and then close the cycle.
Yeah, interesting. Yeah, they're literally disintegrating structures that have been grown, that are sort of bound up, all of this energy and all of this carbon, and they make them useful again through this physical disintegration process.
Yeah.
Interesting. So when we think about the fruiting bodies, the reproductive organs, these things sprout up from time to time, part of the year, and they release spores, and this is how the mushroom network is reproducing itself. And of course, we've got medicinal mushrooms that make things like antibiotics or other nutrients that have medicinal properties. We've got gourmet mushrooms that people or other animals eat as nutrients. And then, of course, we have toxic mushrooms. Yep. So we sort of have this panoply of different options that sprout up on the forest floor. Some of them can kill you. Some of them can nourish you.
Nurture you, yeah.
Why do we see that kind of diversity? And from the mushroom's perspective, why do different mushrooms make these types of things that can affect animals when they eat the fruiting body?
So this questions why do mushrooms, or in more general sense, why do fungi or plants make all these wonderful compounds, be it toxic compounds or whatever. This is kind of the holy grail, the most important question of my research area, natural product research area. And only in very few cases we know why. To be honest, in most cases we simply don't know why an organism invests all the energy, all the metabolic power and multi-step reactions to come up with a particular compound that we think of as a toxin or as an antibiotic. I mean, is the purpose for which this compound, a particular compound, was evolved for? Was it really to make an antibiotic or maybe something else? We use it as an antibiotic because we isolate, we extract, we test, and see bioactivity against whatever. But was this really the purpose of having been evolved? And now coming back to the toxic, in quotes, toxic mushrooms that in textbooks you would consider the magic mushrooms also to be toxic because they interfere with your neurotransmission and cause hallucinations and stuff. So on first glance you would consider them toxic. And this is actually also a question that I I'm trying to answer, have been trying to answer, still am trying to answer, is why was psilocybin evolved? I mean, from part of it, one theory would be you interfere with the neurotransmission of a predator, of kind of a small animal that nibbles on you and tries to feed on you. So this would be a more general concept. This is protection, like other toxins, or what we consider toxins, simply as a protection. And this may be true in many cases, some cases. That they are simply made for protection, so you don't get the producer does not get eaten up, chewed up by an animal.
Mm-hmm. Yeah, one thing that jumps to mind there is when you think about something like psilocybin in the Psilocybe mushrooms, one pattern that seems to be true to me is when you look at psychoactive drugs that come from plants, they typically seem to be made for defensive purposes, to protect the reproductive organ. And I wonder if, do you think, is that a general trend in botany, that a lot of these things that happen to be psychoactive when an animal like a human eats them are actually being used by the plant for defense, to kill insects or to interfere with an animal that might want to eat it?
Tough question. I can't really answer your question. I'm afraid. If this is a general principle, I'd rather think. So what? Let's talk about plants. In case of opium poppy, I would say yes. This is protection. This is my personal guess. So please, all pharmacologists now in the audience, they may look at that differently. So I would say yes. In the case of cannabis, not sure. It's the female flowers, obviously, that are rich in THC. Which is actually not the only compound, which is another question related to yours. Why not one particular, or why in some cases one particular compound? Why in other cases this entire set of many compounds? How many THC related or congeners have been isolated from hemp? I believe 50 or 60 or even more. And so it's not about one particular compound, but more a mixture of them all. So the true ecological reason why they have been evolved is maybe the mixture, not the isolate. We think about from a pharmaceutical perspective or pharmacological perspective, we think in individual pure compounds with defined properties, with defined interactions with a receptor in our brain, but nature maybe thought differently. And now coming back to psilocybin, what I think is the case, and what our research points to. Well, this is. It's hypothetical, but at least an idea to keep in mind is that psilocybin, or the capacity to make psilocybin, has in fact to do with protection, but not by interfering with the predator's neurotransmission, but much simpler, but by just making a protective agent on demand. Think about, you're familiar with that, of course, and many in the audience as well, I believe. with the bluing reaction of the magic mushrooms.
Yeah, when you touch them or you physically bruise
injure them or cut them, they turn immediately. They turn blue, instantly.
Beautiful, yeah, like almost iridescent color.
Mm-hmm. And what I think is the case that the magic mushrooms do not make. I mean, from our anthropocentric perspective, and with this, powerful and wonderful pharmacology that has accompanied, humankind for centuries and millennia even. We think about this such a wonderful phenomenon. This must have something to do. Nature must have evolved this for whatever purpose, to interfere with neurotransmission, or with our brains and our thinking and behavior, our animals' behavior, predators' behavior. But I think that the answer is much simpler. That the mushrooms are not interested in psilocybin itself. It's just a storage form. It's about chemical defense, but it's not to interfere with our brains and our behavior, but to have a compound, an inert compound that can reactivate it quick upon attack, upon demand, or on demand, and then turn into that blue pigment that is that binds to protein, that can precipitate proteins, that kind of can react with proteins and keep predators from feeding on the mushroom. So it's a defense on demand. This is my hypothesis. A defense on demand, kind of a melanin, a protective polymer, and psilocybin is just the monomer that's stable and safe to store.
I see. So the bluing, when the bluing happens, it's a bunch of psilocybin individual molecules that have physically started to stick together?
Yes, indirectly. So what happens? So psilocybin is a stable molecule that you can actually store here in the lab, that can be stored, and it has one very unusual feature. Now a few technical terms will come in. Mm-hmm. It has a very, from a structural perspective, a very unusual feature, and that's a phosphate ester. So a phosphate group is basically sticking on an alcohol group of the molecule. With the phosphate, we call it psilocybin. Without the phosphate, we call it psilocin, the molecule, which is the actual bioactive or neuroactive compound. Psilocybin itself is inactive or little active, but as soon as we ingest the mushrooms, the molecules the psilocybin will be cleaved into psilocin, and that is what causes the pharmacological effects and the psychedelic effects. And psilocin is highly reactive, and it will connect to other psilocin molecules. It will oligomerize or polymerize into that wonderful blue color, and this is kind of the same. And it fulfills the same role. This is my hypothesis. It fulfills the same role than melanin in other other organisms.
I see. So the psilocybin is made in the fruiting body. The psilocybin can turn into psilocin, which happens to be the psychedelic compound that most people are familiar with magic mushrooms. But the psilocins start to oligomerize and polymerize, meaning they start to physically attach to each other, and that's where that bluing comes from. So is this providing like a physical barrier? Is it almost like a physical protection, like a wound healing or something like that? Or is there some kind of chemical deterrent to an insect or something like this?
It's a chemical. It acts on a chemical basis, because this blue oligomer, these psilocin oligomers, multiple individual molecules now being linked to each other, they bind to other proteins say in the intestinal tract of an animal that feeds on the mushroom.
Oh.
And so it's kind of it precipitates proteins, it binds to proteins, attaches to proteins, and then harms a potential predator.
I see. So maybe like the insects literally just get physically sick or something.
Yes, yeah.
Interesting.
They won't continue feeding on it. This is my theory. Yeah.
I see. Okay, so the idea.
Think about a hand grenade. Kind of a strange comparison, I know. It has a pin, that a security pin that keeps the grenade from going off. It's called psilocybin, with the phosphate group still attached. So once you cleave off the phosphate, you pull out the pin, and now it goes off, and boom.
I see. Interesting. Yeah. So the psilocybin has this sort of like little safety mechanism on it. Mm-hmm. When you take that off and you get the psilocin, now you can have this oligomerization reaction where all of these compounds connect to each other, and then you get this bluing reaction that we see, which is just a physical manifestation of what might be this kind of protection mechanism.
Exactly. Yeah.
Is it primarily so from the mushroom's perspective, the fruiting body of a psilocybe mushroom, is the primary predator for this insects, or is it animals as well?
I would say it's insects, or maybe some helminths, some worms living in the ground. Maybe some slugs or like that.
I see. Yeah, slugs,
but not necessarily. I don't. I mean, that this is hard for humans to believe, but I don't think that these little magic mushrooms have evolved the psilocybin for us.
Yeah. One thing that makes that a little tricky, in my opinion, the idea that the mushroom evolved it with animals in mind, is you have to eat enough of the mushrooms to get a psychoactive effect, and of course, there's a delay. So it would make it quite difficult for an animal to learn by association that, if you're a small animal on the forest floor nibbling on mushrooms and other things, it would be very difficult to learn that it was those particular mushrooms that caused some psychoactive effect 2 hours later.
Yeah. But in case of the bluing reaction as an immediate, protection mechanism, or to thwart off a predator. I mean, this happens within seconds. This is an immediate reaction. Interesting.
Yeah, and I know
this is basically related to what chemists call protection groups or protection chemistry. So this is a strategy when you do a synthesis, a chemical synthesis, has nothing to do with nature now. And you want to do a particular reaction at a particular position, but you got multiple reactive positions in the molecule, then you need to protect the others that you don't want to see react, and to just work on one position. And then once you are done with your synthesis, you then need to deprotect, take off the protecting groups again from the molecule. And this is kind of a similar strategy, I believe. This relationship between psilocin and psilocybin, that it really is it's natural protection group chemistry. Because this is something now it's getting a bit more technical. Because I was intrigued by this phosphate ester. Why? Nature doesn't do that very often. This is very rare, and there would have been other options and less energy intensive and less cost intensive options to stabilize a molecule. Why did the fungi phosphorylate? And the answer, I mean, in the light of this of that theory, it's clear, because you cannot just stick something on. You need to make sure that it comes off again when it needs to get out, needs to come off.
So it's protective chemically, but it can easily be removed.
Reversible. Yeah, it's protective chemistry, but it's reversible chemistry, and this is more makes more sense to me.
Yeah, and I guess that would be the perfect kind of protection. That reversible protective chemistry is why that bluing reaction can happen so quickly. You can very quickly change the molecule and let it do what it needs to.
Yeah, absolutely.
Interesting.
And the second thing is, as we basically worked on the biosynthesis, so that the metabolic steps, how the mushrooms actually make psilocybin, is that there's been kind of a proposed biosynthetic route that was devised in the late 60s by brilliant chemists. That they did brilliant work, and they were really, they were ahead of their time back then. And which kind of proposed that psilocin is an intermediate, a precursor to psilocybin. And when we did our research into the biosynthesis, it slowly dawned on us that the mushrooms do not make psilocybin, but they have evolved the entire pathway in a way to avoid, by all means, that psilocybin ever occurs. Psilocin ever occurs. So the complete opposite, and even they even, so this biosynthetic mechanism, step by step, even has a built-in repair mechanism. So should ever psilocin occur in the cell, in the intact cell, not when it gets bruised or something, but in the intact cell. So if ever psilocin should occur in the cell, there's a protection mechanism, a safety switch that protects psilocin again and makes it become psilocybin again. So the fungal cell tries to avoid, by all means, it tries to avoid that psilocin occurs, because it's too reactive. Because otherwise it would exert the toxic effect on
itself.
Yeah. This is kind of, if you pull the pin out of your hand grenade and store it in your garage.
Yeah. I mean, there is kind of an analogy with cannabis here, because the cannabis essential oil, which contains THC acid, it's always put in the trichomes at the external surface of the plant. The plant kind of doesn't want it on the inside. And it actually, at least there's some evidence to suggest that is also offering a kind of, in this case, physical protection, because apparently the THC acid, the acidic group, can absorb photons, apparently. And so it might be, some people think that it might be acting as a kind of sunscreen for the plant, basically, to help absorb some of the UV radiation that the plant is going to be exposed to. So anyways, so the plant is making psilocybin, the fungus is making psilocybin. So these psilocybe mushrooms make this compound, which is very interesting. It's got this protective phosphate group over one part of the molecule. And as soon as you pull that off, that was your hand grenade analogy, grenade analogy.
Kind of brutal analogy, but
but it's interesting.
Conveys the message. Yeah.
As soon as you pull that off, you've got psilocin, which is now very reactive, and it can oligomerize. It can come together with other psilocin molecules, and that offers potentially some kind of protection. But in any case, that's where the very fast bluing reaction comes from.
Yes.
So if we back up into the biochemistry here, can you unpack where the psilocybin is coming from? So what are the starting materials for the psilocybin, and how do you get to that?
Pretty simple. So the starting point is an amino acid that we all have in our proteins, that all organisms have in their proteins. That's called L-tryptophan. It's the heaviest amino acid and the rarest amino acids among the 20 in most cases, amino acids that we basically consist of. And so it begins with with this, general building block that's available everywhere in your food, in the meat or steak or whatever you eat, or plant, vegetarian food. And the first step is that it loses CO2 to make tryptamine out of tryptophan. So tryptophan is what's going in, and the first processing step is that tryptophan loses CO2, carbon dioxide, and produces, or an enzyme does that, produces tryptamine. Step number one. Step number two is another quite unique feature that doesn't occur very often in natural product chemistry, which is, now it's getting very technical. This is that you install an alcohol group, OH, an oxygen atom now comes in at a particular position of tryptamine, position 4. Very good choice by nature to put the oxygen there. I will come back to that later. And this occurs very very infrequently, very rare. This is a very rare feature. And interestingly, it happens as well with kratom. So the mitragynine, the kratom molecule, features a 4-hydroxylated indole system as well. Anyways, so now we've installed the alcohol group, and the next step is the phosphorylation, because now the molecule is kind of unstable and reactive, prone to oxidation. And now the fungus immediately phosphorylates this alcohol group, this 4-OH group, to stabilize the molecule, to basically to push in the security pin. And the final step is, also technical term now, to transfer methyl groups, 1 carbon units, to the molecule, which then completes psilocybin biosynthesis. And this is also one of the questions we were asking ourselves: why this methylation? So it's getting methylated twice. It's not necessary, because it would bind to, the psychedelic effect, or the polymerization effect, the bluing effect, would happen with or without these methyl groups on. And the reason is, again, what I think is the reason, so others may think differently, is that it is required to slow down, to slow down basically, or to make this protection mechanism possible, from psilocin back to psilocybin, this protection mechanism that keeps the cell clear of psilocin.
I see. So
and it kind of helps slow down reactivity to make the protection mechanism faster.
I see.
Fast enough.
So basically, the fungus starts with tryptophan, and it gets it through food, just like we do. So it gets this amino acid from its diet. It then turns this tryptophan amino acid into tryptamine, which is presumably the basic building block of all of the psychedelic tryptamines and other tryptamines.
Exactly.
And you go from tryptamine to psilocybin, and the chemistry there is such that you've got this phosphate group on one piece that is a protection. It's a protection thing to stabilize the molecule, but it's also easy to come off. And you've got these other modifications that make it really easy for the fungus to do this bluing reaction through psilocin, but also easily convert the psilocin back to psilocybin inside of the cell if that needs to happen.
Yes.
I see. So I guess the name of the game here is the fungus. So psilocin, the thing that we is the psychedelic compound that's psychoactive in our brains. It's a very reactive molecule, and the fungus simultaneously takes advantage of that reactivity for the bluing reaction potential protective mechanism to protect itself, but also because it's so reactive, it doesn't want too much psilocin inside of the cells. So there's some other chemistry there that's protecting the fungus from that happening.
It would be harmful to the producer. Yes, absolutely.
Interesting.
Yeah,
and so
you summarized it brilliantly.
Thank you. So that's really interesting. But also, before you mentioned, we talked about cannabis a little bit. It produces THCA, but it also produces this sort of constellation of other things. And there's this sort of, branching pattern of biochemical pathways that get you this cocktail of compounds. Inside of that psilocybin synthesis pathway, is it just sort of one single pathway that goes from tryptophan to psilocybin, or are there other branches that the fungus can use to make other tryptamines as well?
So within the tryptophan to psilocybin pathway, there is almost no branching, very, very minor branching. We discovered one branch, which in very, very minor quantities. So that this is a pretty straightforward and linear thing. But the fungus, the magic mushrooms, other mushrooms as well, other organisms as well, other plants as well, they can make other compounds out of L-tryptophan. And interestingly, the magic mushrooms can make compounds that are referred to as beta-carbolines. Like so the Harmala alkaloids, and they kind of these are also bioactive molecules, these beta-carbolines, but in a completely different way. They inhibit a human enzyme that helps degrade, for example, psilocin. So this is kind of a synergism. The mushroom makes a compound that exerts a certain effect in us or in other organisms, and at the same time it produces a compound that inhibits the degradation of another mushroom compound.
Yeah. So the mushroom, at least certain species of psilocybes, are making both psilocybin and these beta-carbolines, MAOIs.
Exactly, monoamine oxidase inhibitors.
Yeah. And as many people will know, this is kind of interesting because this is, in one organism, you have this cocktail coming together, which is very similar to what the shamans in the Amazon do by combining two different plants for the ayahuasca brew.
Ayahuasca, exactly, yeah. Exactly. That's exactly the same principle. You use a tryptamine and use a MAOI, monoamine oxidase inhibitor, in a cocktail, but the mushroom basically produces it all at once in one organism. But one cautionary note: we published that work, and we wrote it in this publication as well, but it got kind of maybe a little bit in the background. That first off, the beta-carboline contents in the mushrooms are very, very low. I would say too low for true pharmacological effect. That's the first thing. The other thing is the highest, if you want to call them high at all, the highest beta-carboline contents were found in the hyphae, in this fluffy mycelium, in the white thing, not in the fruiting bodies themselves. So the fruiting bodies, they are full of psilocybin, one thing, and the mycelium, the white, cotton wool fluffy thing, is a little bit richer in the beta-carbolines. So they don't necessarily co-occur at the same time at the same place.
Yes.
And this,
The beta carbolines are more prominent in the mycelial network.
In the mycelium, yes. In the growing mycelium towards the tip, we also did some work, or my students and collaborators did some beautiful work to localize that, to localize the contents with imaging techniques, mass spectrometry imaging techniques, and they seem to be localized towards the ends, towards the tips of the growing hyphae. But they don't really co-occur at the same place and at the same time in the fungal life cycle. So this is what went under a little bit in the when people read the publication, because it sounded too good,
Yes, I see.
Yeah, so people, the Psilocybe.
Yeah. People were immediately thinking, oh, okay, this could be why certain mushroom species maybe are more potent, because the psilocin is potentiated by the MAOI. But basically what you're saying is the fruiting body's got all the psilocybin, very little beta carboline content.
Yes.
The mycelial network is the opposite.
Yes.
And it's got higher MAOIs. Is there any hint at what the function of the beta carbolines are in the mycelial network?
Not that I'm aware of. Not that I'm aware of. So this brings us back again to the questions: why are all these beautiful molecules? What are they made for? I wouldn't really. I cannot really tell. At least not in a well-founded way, what these beta carbolines as MAOIs are good for the mushrooms.
Mm-hmm. And now for the Psilocybe mushrooms, what is their ecological relationship to plants and trees? Are they one of these species that is symbiotic with certain plants?
These are degraders. These are degraders, which is actually the reason why it's relatively easy to grow them in your backyard, because their substrate is simple. You need wood chips or just dead organic matter, plant beds that are mulched with these wood chips. So these are simple degraders, or they are even coprophilic, which means that they grow on dung. Which is probably one of the routes how they were distributed globally, that they were introduced, for example, to the or some species that were introduced to the New World simply with cows and cattle, and with the dung then.
I see. And so going back to the psilocybin in the fruiting bodies of these things, is psilocybin uniformly distributed throughout the entire fruiting body, or is it more concentrated in the cap or the stem of the fruiting body? Is there any localization there?
Well, I know that there are investigations, and it seems to be more, to my knowledge, more concentrated in the caps than in the stipes, and the spores basically are relatively free and clear of the tryptamines. But so out of our own research, we haven't really made this differentiation yet. So I can't tell. So we didn't do any research to confirm that.
And so what are the natural types of environments that you tend to see psilocybe mushrooms? I think we just alluded to it a little bit. Some of them grow directly in like wood debris, and some of them are coprophilic, as you said, dung-loving mushrooms.
On meadows, yes.
And so what types of environments, like natural environments, does that mean that they're commonly found in?
Various environments, including basically human-made environments. Just walk through a park. We have that in Central Europe as well, or even cemeteries that are well maintained with all the plant beds that are well maintained and covered with wood material, which is a perfect substrate and provides a perfect habitat for these. For example, one of the potent species that's called Psilocybe cyanescens. So cyanescens, the one that turns blue, the one that develops a blue color. What's the meaning of cyanescence?
Mm.
And it grows, really gregariously in parks and artificial environments. The other one that's so I'm now from a Central European perspective. The other one is called Sema lanceata, a very small one, tiny one, grows on higher altitudes in the Alps, again close to on meadows and past and basically close to cow dung. So this is kind of the habitat, but it's not a case. So the psilocybe mushrooms, it's not a case that you really need to search for in a forest as you would do to pick some mushrooms, some bolides and chanterelles or whatever that are associated with trees in somewhere deep down in the forest. This is not the case with psilocybes. It's more about open habitats.
I see.
Or even on roadside.
Yeah.
On Madison roadsides.
Yeah. So the fact that they're degraders and the fact that human activity often degrades the environment, we break up trees to make mulch and things. They like those environments. So is there, there's been some speculation that, humans and mushrooms, psilocybes may have co-evolved in this sense that, actually humans create so many environments that are the right environment for some of these degradative mushrooms to grow, that through just us moving around the world and doing human stuff like making paths and gardens and stuff through the woods, that we've actually facilitated their dispersal throughout the world.
We facilitated dispersal, but I wouldn't call it a co-evolution, because the mushrooms have been around for many more millions of years than we humans do. So I'm a bit skeptical that it's really a co-evolution, but we basically created environment that helped them grow more abundantly, and it brings us back to the anthropocentric perspective. If there's a mushroom that will never get extinct, it's psilocybe, right? That's kind of
yeah. And
same with cannabis, with hemp. Think about a plant that humans really care about and care for. Then it's probably hemp.
Is it so when you're studying these fungi in the lab, how advanced are the genetic and molecular techniques? Like, can you make transgenic mushrooms?
No. It has to do with the life cycle of these mushrooms, which is a little bit complicated from a cell biology perspective. So the answer is no, or say not yet.
I see. And one of the things I think is
in general, so mushrooms generally are pretty are not easy to manipulate genetically. There are some techniques, but we've never applied them, and at least not successfully, to the magic mushrooms.
Mm-hmm. And how abundant is psilocybin or psilocybe mushrooms in nature? Like roughly how many species of psilocybes are there? And is psilocybin produced anywhere else in nature? Is it only the psilocybe mushrooms, or does it show up anywhere else in other fungi or other plants?
Not in plants. So it's so the number of species about psilocybe and other genera, very roughly, I would say 200 species globally. Mm-hmm. Very roughly. It depends a little bit on taxonomical issues, because sometimes one species is split up in 5 or many species. Sometimes they are then consolidated again back to one species. So it varies a little bit. I would say roughly 200 species. And where else does it occur? Definitely not in plants, although plants are very talented at and good at making tryptamine bioactive compounds, but not psilocybin. And there's some there have been some reports that psilocybin can also made by say basal fungi, which still need to be confirmed.
I see.
So mostly basically it's about these higher higher mushrooms.
So mostly found just in these psilocybe mushrooms.
Yes, yeah.
But there's quite a few species depending on how you count.
Mm-hmm.
Interesting. And now, what about, are there any other compounds that have interesting physiological properties for humans that are related to psilocybin? Some of these other alkaloids that I know that some species produce, basis-stin, norbasis-stin. What do we know about some of these other compounds, and how common are they?
So biocystin, norbiocystin, these are intermediates on the way from that we just talked about, from tryptophan to tryptamine to psilocybin. So they are just intermediates, and they occur in variable amounts. It differs a little bit from species to species, and so research from what I observe, research into their role, qualitative and quantitatively, has just begun. So we don't really understand yet the contribution, or at least maybe that there may be some literature, but I'm in the moment I'm not aware of any. So we only slowly begin to understand what role these other compounds play for the pharmacology, because from a pharmaceutical perspective, we think in pure compounds, in homogeneous pure compounds with a reproducible ligand, as a binding molecule, ligand receptor interaction. This is what we can measure and quantitate. And I mean, there have been reports related to magic mushrooms on the so-called entourage effect that other compounds may affect and increase or decrease the action of psilocybin. There's this mysterious wood lover's paralysis.
Can you explain that for people who don't know?
Absolutely, sure. It's a pleasure. So these are reports. So wood lover's paralysis. These are reports more on an anecdotal basis, not really in the scientific literature, or to a very small degree in the scientific literature, that people who consumed magic mushrooms as a recreational drug experienced paralysis, kind of numbness, or really they were heavily impaired in coordination of their muscles, and these are all and these effects occur with those mushrooms that grow on wood. This is what called wood lover's paralysis, with the wood inhabiting mushrooms, not with the ones that grow on dung, like Psilocybe cubensis or Sema lanceata. And so this was kind of or it still is kind of miracle. What causes these paralytic effects that we cannot move our arms anymore, or feel numbness and so on. And because the effects, that these paralytic effects, that last much longer than maybe 18 or up to 24 hours, much longer than the psychedelic effects of psilocybin, which is maybe 3 or 4 hours until they subside. And this is not number one. Number two is that that psilocybin causes central effects on our central nervous system, whereas the paralytic effects affect our peripheral nervous system. So that there must be something different going on, but we don't know why. This is a miracle. And try to has not much been research been done yet. And which kind of sounds like there are other compounds, at least in some species, some particular species of mushrooms, that are not discovered yet.
I see. So there are anecdotal reports that some of these wood-loving psilocybes, in contrast to the coprophilic ones that grow in dung, you can get this paralysis that far outlasts the psychedelic effects. So there's probably some other chemistry there, but we just don't know what it is yet.
Don't know yet. Yeah.
Interesting. So what other stuff do you work on in your lab? It looks like you also work on things like terpenes produced by fungi. So what are some of the other families of compounds that your group is looking at?
The terpenes are not a focus in my group. It's more about and are we talking about the magic mushrooms or the other projects that are going on?
Anything else.
Okay, because I mean, the magic mushrooms are one project, maybe the most visible one in my research, but surely not the only one. And there are others. For example, with now we move from the magic mushrooms to the web caps, symbiotic mushrooms. So we work on another group of natural products, which are called polyketides, which are distantly related to fatty acids. Also a very prolific group of natural products, many bioactive molecules. So this is another focus. How they and we recently found out that an undiscovered class of enzymes actually make these mushroom polyketides, or these oligocyclic mushroom ketides. So this is another focus, not necessarily the terpenes, although I really like the terpenes, and it brings us back to the spices and herbs. In the beginning of our conversation, I really like to spice up my dishes with thyme and stuff. So the web caps, this one thing, and and another type of compounds are that brings us back to colorful and colorful molecules, are a group which is called pulvinic acids. It has nothing to do with psychoactive molecules, but they seem to play a role in interactions between bacteria and fungi. So they impact upon biofilm formation and stuff. And so this is so this terpenoquinones, these pulvinic acids, this is a group of compounds that I've worked with for many hours, which is basically the one that I then embarked on when I thought about a postdoc, which program should I develop as my own independent program 15 years ago. And this is still active in my group. So it's basically the three things. It's the magic mushrooms, it's the polyketides, mushroom polyketides, and the pulvinic acids, which are very frequently found in boletes, in bolete mushrooms, and which are the molecules. So when you again, this is a phenomenon many mushroom enthusiasts in the audience will be familiar with. So when you bruise a bolete, it will turn blue. Has nothing to do with the bluing reaction of the magic mushrooms. It's a different chemistry. And these pulvinic acids, my third project, is about these compounds.
I see. So there are mushrooms that will bruise blue, but they're not psilocybin mushrooms.
Exactly. Yeah.
Well, that brings, to maybe a practical question. There's a fair number of people in the world that like to go mushroom hunting. I live in the Pacific Northwest. I buy a lot of mushrooms at the market that people go out and they just pull right from the wild. And, this can be, if you're in the right environment, potentially dangerous, right? Because you might pick something that's toxic if you're not a seasoned mushroom hunter. So for a psilocybe mushroom, if someone's hunting for magic mushrooms containing psilocybin, what are the characteristics that they look for that would distinguish it from other things that might be harmful, for example?
Well, bluing is one feature, but not all mushrooms that, are we talking about the edible ones or the psychedelic ones?
The psychedelic ones.
The psychedelic ones. Okay, so the psychedelic ones, they should bruise blue, but not all mushrooms that do so are psychedelic. So it's not working in that direction. So the psilocybe mushrooms are well, you need to be an experienced mushroom hunter if you want to pick them for whatever reason as a recreational drug. I shouldn't, this is not something I recommend or endorse, by no means. There's also legal restrictions in some states. But just from a mycological perspective, the psilocybe mushrooms are quite variable in the way in their appearance, in their morphology. From cubensis, which are not very likely to find in the Pacific Northwest, which are really tall mushrooms, down to tiny. Tiny species. So there's no real, one feature fits all trait that would help you identify. With the naked eye, that there are microscopic features that then help you identify a certain species. But for the cyanescence, which is basically one of the widely distributed wood-loving mushroom, it has what's characteristic is that it grows gregariously in clusters, in basically in troops, and the margin here turns a little bit bluish as the fruiting bodies age, and they are kind of a wavy shape here. It's not as straight as here. But it's kind of...
So basically, it sounds like you're saying that all psilocybin-containing mushrooms should bruise blue, but just because something bruises blue doesn't mean it's a psilocybin mushroom.
It's a psilocybin mushroom. Yeah.
So it needs to bruise blue, and you also need to probably have good familiarity with the specific morphology of the species that is likely to grow in your area.
Yes. And plus, to make it more complicated, there's a lot of psilocybe related mushrooms that do not make psilocybin.
So they morphologically look similar, but they don't have
look
psilocybin.
similar, but do not make psilocybin. They have been from a taxonomical perspective, they have been separated or grouped into a different genus. Now, so psilocybe mushrooms are now, from a taxonomic perspective, now are required basically to make psilocybin to really belong to that particular genus. But there are many out there, and this is also the reason why say you have really don't take any chances, because similar looking mushrooms can be very, very toxic and make some. I'm not familiar or not familiar enough with the microflora in say in North America or the Pacific Northwest, but at least from the European one, I can say that similar looking mushrooms make the toxin that also makes the death cap toxic and fatal, basically.
What is that molecule?
Ammonidin, which basically attacks your liver tissue and blocks processes in your liver, inhibits your liver, and which eventually may cause death. And what most people don't know is that not just the death caps make ammonidin or ammonitins. It's a family of compounds, but also other ones, and among them, little brown capped mushrooms that may, at least at first glance, look similar to psilocybe, for example, cyanescence.
I see.
And unexperienced, unexperienced people may really confuse them.
Yeah. So I guess to kind of recap what you've told us about psilocybin, there's really this one genus of mushrooms with maybe some minor exceptions, but it's the psilocybes that produce psilocybin. It's produced in the fruiting body, in the reproductive organ of the plant. It's got this interesting biochemistry where the fungi are eating tryptophan in their diet. They're turning tryptophan into tryptamine into psilocybin. And you told us about this interesting chemistry where the molecule is stabilized and protected in certain ways, but it's done in this reversible manner, so it can very quickly make the psilocin bluing reaction happen, plausibly as a way to protect itself from things that want to eat the fruiting body. And it evolved that biochemistry for its own reasons, and it just happens to be psychoactive when an animal eats it. Yeah, it's a very, very interesting organism. What do we know about like the genetics underlying this? Are there particular genes that evolved in this lineage of psilocybes to allow it to have that biochemistry?
So basically, we need 4 enzymes to convert tryptophan into tryptamine, into norpiocysteine, biocysteine, psilocybin. And these 4 enzymes correspond to 4 genes, which are present in more or less the same arrangement. Maybe sometimes some duplication happen in the psilocybin mushrooms, psilocybe, and also in some other genera. So it's not just psilocybe. I mean, although they are most familiar and well known, but there are also some other genera, and that do contain a kind of a genetic locus, a portion of their genome. 25,000 roughly 25,000 bases are necessary, that contains these 4 or sometimes duplicated 5 or 6 genes to make psilocybin happen. And there's one study that came out almost 5 years ago, not out of my lab, a colleague of mine, Jason Slott in Ohio, who basically elucidated how these genes found each other, and outside the genus, and then the psilocybe, and then how the entire cluster of genes got transferred and then radiated out. Beautiful work.
Interesting. What kinds of for the psilocybin group in your lab, what are some of the questions that you're working on today? What's on the cutting edge that's top of mind for your group right now?
It's the bluing reaction still, which is kind of one focus. It's also about the enzymes, to characterize the enzymes in greater detail. For example, this protection mechanism to bring psilocin back to psilocybin. This is something we still focus on, and so it's still active project.
Well, you've shared a lot of interesting stuff with us, Dr. Hoffmeister. Are there any final thoughts you want to leave people with about, the biochemistry of psilocybin mushrooms or mycology generally?
Some final thoughts. It's maybe on a more general thing that I've. We talked a lot about science and discoveries and theories, hypotheses, which would not have been possible to discover without very, very dedicated co-workers and group members here. So I would like to mention Alex Sherwood of the Hans Knöll Institute, Madison, Wisconsin, who is a fun guy and so such a pleasure to work with. It's with Andrew Shadeen of Chemtech, who actually appeared on your in I think it was episode number 71, who is also it's so inspiring to share thoughts with him and cooperate with him. With Bernhard Roop, he's an American-Austrian protein chemist, also fun guy, and so they all. I'm really privileged to cooperate with all of them, and develop hypotheses and share thoughts and get input and provide them with input, and also with a very enthusiastic group here, a very dedicated group here behind this wall. Who actually do the creative work and the lab work, and really come up with new knowledge. And so this is really a privilege, I think, and which I would like to emphasize. That's not just one person who's standing in the front, but it's about the cooperators and the co-workers that do all the work and. So just like the mushrooms, it's a lot of symbiosis. Exactly. Yeah.
All right. Well, Professor Dirk,
and one that's very, very well working.
Yeah.
In my case, fortunately.
Professor Dirk Hoffmeister, thank you for your time, and I look forward to talking to you again at some point.
It was a pleasure. Thank you for having me.